Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Alberto Serrano-Pozo, Assistant Professor of Neurology at Harvard Medical School and a staff neurologist at the Memory Division of Massachusetts General Hospital. Alberto is also a 2022 Toffler Scholar. Today, we will be discussing how Alberto and his team are researching the role of glia in Alzheimer's disease. So welcome, Alberto.
Thank you so much for having me.
I'm looking forward to this conversation. You're doing some really interesting science, and I'm excited to kind of share this novel kind of approach to this disease. Can we just start with, in general layman's term, what is the focus of your research and your lab?
Sure. I have two primary areas of research. One is the role of astrocytes in Alzheimer's disease and other dementias. Another one is the role of APOE, one particular gene in Alzheimer's and related dementias. So astrocytes are these cells that we all have in the brain. We have the neurons or nerve cells, and then we have another group of cells which are non-nerve cells. They're called glial cells. And one of those types of glial cells are the astrocytes. They're called astrocytes because they have this star shape. They have, a cell body and then multiple branches that make them look like stars. And they are very intriguing. We've known for a long time that they are very important for the normal functioning of the brain. And we know also for a long time that they go awry in Alzheimer's and other dementias. But we don't really understand yet whether those changes that they undergo in the disease are good or bad for the brain. So that's one area of primary focus. And the other one is this APOE gene, which we've known for a long time, almost 30 years, or more than 30 years, that is one variant of this gene increases your risk of having Alzheimer's disease. And the gene is primarily expressed by astrocytes precisely in the brain. So we think it's involved in many of the changes that astrocytes undergo in Alzheimer's. And recent research has shown that it's also involved in many of the changes that other brain cell types undergo in Alzheimer's. So it's always been a hot topic for Alzheimer's research, but recent discoveries have put it at the center stage of Alzheimer's research.
What does that mean for something to express something? I think you said that the astrocytes express the APOE. What does that mean?
So, every cell type has this gene in the brain or elsewhere in the body, but some cells have the ability to convert that gene into messenger RNA, and then that messenger RNA can be or not translated into protein. The proteins are actually the molecules that have the effect on the cell function, and they are kind of the effectors, the executioners of the gene message, sort of. And even though all cells have this gene in their genome, in their DNA, not all cells express it at the protein level. Not all the cells convert the gene into a messenger RNA and the messenger RNA into protein. In the case of the APOE or apolipoprotein E, which is how it's spelled out, the astrocytes are the main producers of these, and they secrete it outside the cell, and then it's kind of this APOE works as a messenger for other cell types. So the astrocytes influence other cell types in the brain through secreting this APOE protein, and that's why what happens to the astrocytes, we think, can affect the whole brain functioning.
So I think you told me once that this is kind of a new area, just focusing on astrocytes, and I believe you said you're really concentrating on a part of the brain that really hasn't been explored too much with these astrocytes. Am I understanding that correctly?
Yeah, that's exactly right. So for many, many decades, the research on the brain has focused on neurons, on the nerve cells, and has kind of put aside the rest of the cells. But thankfully, in the last few years, I would say in the last decade, there's a growing interest in investigating each of the other brain cell types, and astrocytes among them, and not only from the point of view of the normal brain, the normal functioning of these other cells, but also how they go bad or how they change during different diseases, and what are the contributions to the development of different diseases and the progression of those diseases. And in particular, astrocytes are getting more focus, more interest, because in the normal brain, astrocytes are sort of the housekeepers. They make sure that the nerve cells work well. They fine tune the activity of the neurons. They make sure that the blood supply to the brain is coupled with the neuronal activity, that if our brain is more active, then there's more blood supply. If our brain is less active, if, we are sleeping or something, then the blood supply goes down. Then the supply of nutrients and glucose to the neurons is regulated by astrocytes. There are many other functions that are important for the normal activity of the brain depend on the astrocytes. So it makes sense to study how they are affected by disease, because if we understand how they get sick, maybe we can fix them and then improve the general functioning of the brain, slow down the progression of diseases like Alzheimer's.
I think it's maybe more common for people to have heard about amyloid plaques or, these tangles in the news, especially with some of the drugs that have come out. What is the relationship between these cells that you're studying, the astrocytes and such, with the amyloid and the tangles?
Great question. So, in fact, what we see under the microscope when we look at the brains of our patients who are generous enough to donate their brains for research, we see that these astrocytes change their shape and change their molecular makeup is precisely around the amyloid plaques and around the tangles. So we think that the amyloid plaques and the tangles not only affect the neurons that are surrounding them, but they also have a negative impact on the astrocytes, and that the astrocytes are reacting to them to try to do what they do best, which is protect the neurons. But we also think that somewhere along the progression of the disease, this astrocyte reaction to the plaques and the tangles may turn bad, may turn toxic, they may start causing harm on the neurons, perhaps by releasing inflammatory molecules and neurotoxic molecules that may actually add more fuel to the fire of the plaques and the tangles. So those processes are the ones that we are trying to study using mouse models, using also in vitro cell models, and manipulating some molecular targets to see how that affects the plaques and the tangles.
So if I understood that correct, it sounds, or at least there's a hypothesis that the amyloid is affecting the astrocytes. And if the astrocytes are the keeper of kind of the neurons and kind of good cellular function, that it just overloads it and maybe kind of breaking down the process. Is that kind of what I understood?
That's exactly right. Yeah, we and others have found that the astrocytes, in response to the plaques and the tangles, they gain some toxic functions that they didn't have in the normal brain, and they lose some of their normal functions as well. Those protective functions that they normally have in the normal brain are lost. We know that because we and others have analyzed precisely the messenger RNAs that they contain. Thanks to technological advances, we've been able to isolate the messenger RNAs from individual cells from the brain of patients with Alzheimer's disease and normal control brains, and have been able to understand what are the molecular changes that these astrocytes go through in the disease, and have found that many of the messenger RNAs that encode proteins that are important for the normal function of the astrocytes are decreased in Alzheimer's astrocytes versus control normal brain astrocytes, and some other genes that we think are deleterious for the brain functioning are increased in the disease astrocytes. So that is kind of a perfect storm. If imagine, if an important cell that normally has a housekeeping role stops doing the housekeeping, but on top of that, it starts, misbehaving and expressing toxic proteins and releasing toxic proteins into the environment, it could potentially poison other cell types and particularly contribute to neurons, to the demise of neurons. So that's why we think it's so important to study them.
So you mentioned APOE, and I believe there's different kinds of these proteins. There's like APOE4, maybe APOE2. You study these also. Can you expand on what the difference is and what you're seeing?
Sure. So APOE is a gene, it stands for lipoprotein E. And originally, what this APOE is thought to do is to transport cholesterol within the brain. We all have to repair the membranes of our brain cells, and those membranes are made of lipids, fat, and we need a protein that transport fat between brain cells so that those membranes are repaired. So that's the main function of APOE. And as you said, there are three variants of this gene and three forms of the protein that this gene encodes. There's the APOE2, APOE3, and APOE4 variants. And the APOE4 is the one that increases the risk of Alzheimer's disease. We know that since 1993, when this association was found, people with the APOE4 variant have an increased risk of developing Alzheimer's, and people with the APOE2 variant have a reduced risk. They are protected from developing Alzheimer's, as compared to people with the APOE3 form, which is the most common in the general population. This is sort of the textbook definition of APOE, but we've learned so much in the last decade about APOE that the dogma that I just said is being shaken right now. For example, recent research has shown that APOE4 may not be as bad in certain populations. It looks like in Blacks and Latino people, the APOE4 is not as strongly associated with Alzheimer's disease risk as in the non-Hispanic whites. There's a lot, or in the Asians, and non-Hispanic whites, the risk of Alzheimer's associated with the APOE4 variant is very, very high, but not in Latino and Blacks. And we don't understand yet why. That's an area of active research. If we find out why this difference is happening, we imagine we could try to target those mechanisms in order to reduce the risk associated with the APOE4 variants in Asians and non-Hispanic whites. So another major advance in the last decade is we thought that the only mechanism why APOE4 increases the risk of Alzheimer's is by increasing the accumulation of amyloid in the brain. But recent research by many groups has shown that the APOE4 variant essentially changes the behavior of multiple cell types in the brain, not only astrocytes, but also microglia, which is another cell type, oligodendrocytes, which is another cell type, endothelial cells, which are the cells that line our blood vessels. So it has multifaceted effects in pretty much every brain cell type. That makes APOE all the more interesting. And that's why it's really mysterious, because it is a very small protein. We know it transports lipids in the brain, but there's more and more evidence that it acts as a messenger between cells, and it has these distant effects in other cell types that don't produce it that much, but they receive it from the astrocytes or the microglia, which are the main producers of this.
So you mentioned APOE2 may be kind of a resistor and provide resistance. Is that common across the different demographics too, or is that being kind of shaken?
Fortunately, APOE2, which is the protective variant against Alzheimer's, is the least common. Yeah. So one area of active research, and actually close to being tested in clinical trials in humans, is converting APOE4 into APOE2, converting the risk variant into the protective variant. They only differ in a couple of amino acids. So there are new technologies that are trying to convert one into the other, so that we can protect people from developing Alzheimer's disease, people who are a higher risk because they carry the APOE4 variant. So that, obviously is being tested first in mouse models, and then, in maybe non-human primates, and before getting into clinical trials in humans. But, we are hopeful that this strategy with the advances in gene editing will become a reality someday.
So not only are you a researcher and you run a lab, you also see patients and you're a doctor. You're also, you must not sleep. I don't know how you do that. But how do you integrate these findings, your research? How do you do that back and forth? I mean, what's a typical week for you, and how do you do this kind of dual role?
Yeah, so it can be challenging, but I do see patients with memory disorders once per week, and then do research the rest of the time in the lab. It is a two-way street. I think my work in the lab helps me with, in the clinic, being inquisitive with my patients with differential diagnosis. When I'm in front of patient and trying to figure out what's going on, I'm thinking, besides the human aspect of the disease, I'm also thinking about the plaques, the tangles, the astrocytes. I'm kind of imagining what's going on in that brain. I just I don't look at brain MRIs and so forth, but also I'm thinking about the brain at a cellular level, at a molecular level. So that is kind of very enriching. And on the other hand, there are questions that come up in the clinic that sometimes they actually open new avenues of research for me in the lab. And there are several examples of that have inspired me. I found inspiration in the clinic. I wonder. Then I went to the lab to try to figure out whether the hypothesis that I came up with in the clinic was right. For example, one theme that is kind of obsessive is why some patients in the clinic decline faster than others. Why there's this huge heterogeneity in the rate of clinical progression of Alzheimer's and other dementias. And we and others are trying to model that heterogeneity in the lab, and whether it's genetic differences that influence the way the brain cells behave and respond to this pathology, or whether they're acquired environmental lifestyle habits that the rate of progression is really a mystery. And some of these aspects are difficult to model in the lab, but we still scratch our heads and try to come up with ways of testing some of these factors in the lab.
How do you choose, or how do patients come in to you? Is there a pretty strict screening before they're kind of assigned to you, or how does that work? Because with you being a researcher and a clinician, do you take whoever comes through the door, or is there some sort of criteria?
Typically, most patients are referred by their primary care physician. They first go to the primary care physician and report changes in memory, and then the primary care physicians refer them to us. But it is also possible some patients are self-referred. We do have some criteria to accept the patients based on age. For example, some memory disorders that occur can be due to traumatic brain injury, or other injuries to the brain. Our clinic is very specialized in neurodegenerative disorders of the age related. So we usually don't. There are other clinics at MGH for memory disorders that are due to acute injuries to the brain, whether it's TBI or stroke or something along those lines. So we do have some criteria for selection, but the main sources of referral are the primary care physicians, and also self-referral with some screening process involved.
So, Alberto, just kind of going back in time, how did you even decide to choose this as your profession and your professional goals, I guess? Like, how did you get into this?
Yeah, it was the decision-making process obviously goes in sort of stages or steps. I knew since high school that I wanted to do research. I knew that since I had the biology class, and my teacher taught me how the double helix of DNA was discovered, and I kind of fell in love with science right there. Then I decided to go into medicine because I thought, maybe I like patient care, and maybe I like the human aspect of this. But I still want to do research. I'm going to medicine just to do research on diseases. Then it turned out that I enjoyed patient care, patient contact. Trying to help patients and families, and I didn't know still what specialty to do after the medical school. And I thought, okay, maybe I should go into oncology because there's a lot of research going on in cancer. But if not oncology, maybe I should do maybe neurology, because neurology, besides the research, there's a lot of it's a very rich specialty in terms of differential diagnosis and neurological exam, and so that's how I decided to go into neurology. And then once you are in neurology, of course there are multiple subspecialties, and I decided to go into Alzheimer's and dementias. I soon realized during my residency in neurology that there was no cure for any of these diseases, and there was a lot to discover. And then I realized also that I have I have strong family history. I remember that realization. For some reason I didn't go into neurology knowing that, but I remember my first year of my neurology residency. I connected the dots, and then I said, okay, I need to other subspecialties are interesting, but I feel the call to do this.
Yeah. Yeah.
And
be a part of the solution, right? It's a personal driven.
hopefully, yeah.
Yeah.
But at least I feel like I'm doing something to help.
Yeah. You talked about the last 10 years, some of our assumptions or some of the hypotheses about what's going on is changing. You and I have talked a lot about kind of the rate of change and such. What do you see, like just even, on the horizon with new technologies? And you mentioned gene editing, you know, are we accelerating our knowledge? Are we getting closer to understanding? I mean, give me a sense of what you think the next 5 or 10 years might hold for us.
Absolutely. I think, I've been doing research on Alzheimer's for 15 years, including my postdoc, and I can see the acceleration of the pace. I have enough perspective to see it, and there are multiple parameters that can attest. For example, the number of journals, the number of publications, the knowledge that is being produced. Is impossible to apprehend, everything that is going on, and then the technological advances that are happening. It's incredible. It's a very exciting time to be a researcher, and AI, I think, is we just witnessing the beginning of a revolution, and it's gonna help to make sense of all this knowledge, and maybe even generate new hypotheses that we didn't connect the dots before, and we didn't think of, and so I think, obviously, I hate when people give a sort of a timeline of when we'll have the next breakthrough or the next drug. I don't like to give false hopes to people, especially seeing patients and understanding what they go through and the families go through. I don't, but there's definitely an acceleration of the pace of research, and there's more collaboration than ever. There's more sharing of knowledge and data sets, and tools, resources. If we keep pushing, we'll get there. During my lifetime, hopefully.
Yeah, hopefully. Tell us about your lab. Like how many people, what's a day to day, and how do you add kind of that leadership to your plate? Because you've got to be there mentoring and kind of coaching them through things too. But what does that look like?
Well, that's mentoring is one of the perks of being a PI, of being a principal investigator, having a lab, mentoring these young kids, and seeing them grow and develop their careers and move on to the next step. Is one of the reasons that keep me going. And I have a small lab right now. We're in transition from people who left last year, and new people who join. And I think we have a very nice environment. We meet every week. We go through raw data and discuss, brainstorm. The lab meeting is like a brainstorming session where we promote free speech. Everybody can, and everybody should speak up and bring up ideas and solutions to the table. And I believe in lifelong learning. There's no hierarchy when it comes to knowledge, and I believe that if you are older, chances are that you know more than the younger. But these kids are really smart. They know much more than me in many things. So I think it's bi-directional, and so we have fun learning together and teaching each other, and when we discover something, even a small thing, we are all happy. It's like a team effort, and especially with if you promote this collaborative environment, then one person's discovery is everybody's discovery. It's kind of fun.
You had told me one time, because we've known each other for a while now, but you told me one time that, and this goes back to the change of just how fast we learn things, that you had a textbook maybe 10 years ago, 15 years ago, that said something like the brain is its own organ and it doesn't, there's not much interface with the rest of the body, too much. I don't know exactly how you categorized it, but, you could kind of share that. And then really in the context of what is the most kind of surprising thing from, when you started 15 years ago to now, because I find that very surprising on how fast things are changing.
Yeah. So if you're asking me whether there's any example that of a dogma that was kind of debunked, that's a good one. Let me think. Yeah, I think in general, probably one of the main in Alzheimer's research, one of the main shifts that have happened is this idea that the brain is not just the neurons, and that every single cell type seems to either have an active role in the disease development and progression, or be secondarily affected by the disease pathology. So this idea of studying each cell type of the brain, and individually, and also this idea of investigating how alterations in one cell type affects each of the other cell type is like, we are thinking more and more of the brain diseases, in particular Alzheimer's, as a dominoes effect, where you the different brain cell types are not independent from each other. This is kind of it's a very simple concept. Obviously, it's so obvious, right? That the brain works as a fine-tuned network of neurons and astrocytes and that all these cell types work together to produce actions, thoughts, language, etc. And that, in any given disease, but particularly in Alzheimer's disease, if one goes wrong, the others can also be, affected. And this idea of figuring out, what is the primary alteration, and then what are the collateral consequences of that alteration in the other brain cell types, and this idea that this vicious cycle, that can we develop therapies that are targeting each of these brain cell types, like can we consider Alzheimer's like cancer, where you have multiple therapies targeting different, you have these cocktails of chemotherapy drugs, each of them is targeting one particular alteration. Can we someday have this cocktail of drugs, one to remove the amyloid, one to remove the tangles, or prevent further tangle accumulation, but then also one to cool down the astrocytes, one to cool down the microglia, and that way try to break this vicious cycle. And I think this interest in the other brain cell types that are not neurons is fairly recent. I would say the last decade, and it can get us very far in terms of understanding how all these pieces fit together in the brain, and how we can target these different cell types with very different but complementary drugs that could work synergistically to improve the health of the brain. That's sort of that approach. I think it's kind of a paradigm shift that we are witnessing.
What do you wish people knew about what you are either doing or studying that they probably don't know, and you wish they did know?
That I'm studying in my lab.
Yeah, studying in your lab, or just the challenge of being a researcher slash clinician?
Yeah, I wish I knew, but I don't regret it, But I wish I knew before getting into this how to switch from one hat to the other, and it's not just those two hats that we wear, the clinician and the researcher. We also wear the writer hat, if you want. We write grants, we write grant proposals, and we write obviously papers, manuscripts. The talk the deliver hat, the speaker hat, which I'm not very good at, as you can tell.
Wow, you're great.
The admin hat, we had a lot of administrative work to do as well. It's like a small business that you're running, and I'm not very good with numbers, and, all these hats that you have to swap, and in the same day you may have to do two or three completely different things, and that is kind of that makes it more challenging, and it's not that you get that's not taught in many of these things are not taught. They are not, you cannot find in textbooks, or in these skills sort of come, you learn by experience, by the hard way, and that's sometimes can be challenging, but the fun in the lab, and the work in the clinic really pays off. The all the other stuff that I didn't count on when I when I decided to take this path.
No, I appreciate that. I think I do think there is a chasm between, I think most people know that we need science, and the importance of science and what science does for us. But I think there's a lot of people that don't really understand the process of science. And it's people like you that you don't sleep a lot. You don't have many vacations. It's not like you're just going into a lab and tinkering. It's pretty intense. And so I appreciate you kind of you sharing that. We kind of talked a little bit as we're wrapping up here. You talked about some of the acceleration and your hope. Is there any kind of one thing that you really hope, for the future? The obvious would be that the disease would be, you know, annihilated, right? But, just from your perspective, what do you really hope for the future?
Yeah, I'm uh, I'm hopeful that the new drugs, that have been approved, they that they that they demonstrate to be the beginning of a new era in which there will be new drugs that will add on into this progress, and that this will be the first of many steps that will lead to at least slow down the rate of decline of Alzheimer's, and hopefully arrest the progression, if possible, revert it or even prevent it. But I think we're going to hear in the next few years of new breakthroughs, through in the form of clinical trials showing the efficacy of these drugs, and of new drugs that target the tangles, and hopefully also down the road, although they're lagging behind, drugs that target the astrocytes, the microglial inflammation in the brain, etc. And then we will have this cocktail of drugs that depending on your biomarkers, depending on whether you have evidence of not only amyloid plaques and tangles, but also astrocytes being too reactive, or microglia being too inflammatory. We could offer a personalized therapy plan, and monitor with biomarkers the response to those drugs, and obviously that's the goal. It's going to take a lot of effort, a lot of a lot of energy and a lot of heads put together. But if you look back, there are reasons to be optimistic. I use the term reserved optimism, or I don't know if that's the right translation, but like cautious optimism, if you wish. There are reasons to be optimistic without, giving false hopes to anybody. But I do think that this is a powerful enemy. We need to understand its weaknesses to tackle this problem, and we need to do it from different angles. It's not going to be a single drug. It's going to be multiple molecular targets, multiple drugs directed to those targets, and each with one mechanism of action and one goal. Hopefully, these new drugs that were just approved in the last couple of years will be just the beginning of the end of Alzheimer's. And definitely, we are working hard. There are many great researchers doing great science, and of course, the pharma companies are also very interested in this, and we should remain hopeful.
I appreciate that very much. So I know I've taken up a lot of your time, but Alberto, how do people get in contact with you if they have questions, if they want to collaborate, if they have money? Like how would they get a hold of you?
Sure. So I think I'm in LinkedIn, in X, and of course, happy to answer any questions in my free time.
As always, Alberto, I really appreciate your time. I learn from you every time I get a chance to speak to you, and I appreciate you spending this time with us. I know you're really busy, but it was very, very enlightening. Thank you very much.
My pleasure.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.